Hello, I'd like to tell you the difference between NGS and Sanger sequencing. So, in principle, the concept behind Sanger sequence and the next generation sequencing are similar. In both cases, in NGS and Sanger sequencing, we add the DNA polymerase into the fluorescent nucleotides one by one onto the growing DNA template strand. And then after each incorporated nucleotide is identified by its fluorescent tag. So therefore, what could be the main difference between Sanger and NGSs? So the main difference goes into the sequencing volume. If we have Sanger sequencing, we only sequence a single strand at a time. So if we need to sequence DNA, we only sequence a single strand at a time. But if we need to do thousands of or millions of sequencing of DNA fragments segmentlessly uh, per run, so in that case we need to use NGS. So NGS is definitely, it is a massively parallel sequencing millions of fragments simultaneously per run. So therefore this NGS is high throughout the process into the sequencing hundreds to thousands of genes at one time. So we can sequence a lot of genes at a time, right? So the NGS, they offer a great discovery of power to detect a novel or if there is an rare variant with a deep sequencing. So therefore, we have deep sequencing in case of NGS, right? So while we are doing a deep sequencing, we may have we may have the chance to get rare variants or just novel variants. That means we detect some pathogens or some mutations that has never been reported before. So in that case, NGS is very important. So it is powerful not to do such kind of things with a deep sequencing. So therefore, when we say NGS, that is just the massively power sequencing technology. So that means we can sequence thousands, and millions of genes at one time, right? We have said already about that. So therefore, the massively power sequencing technology that's called the next generation sequencing has revolutionized the biological science. How? With its ultra high throughput and its scalability is very high, and also its speed it's very high. It has a high speed. So therefore, NGS is most of the researchers are able to do a wide variety of applications with NGSs, and they study the biological system at different level, which never been possible. So by now, NGS is a good technology that we use for such kind of purpose. So therefore, today, a complex genomic research question demands a depth of information behind the capacity of the traditional DNA sequencing. So therefore, we have said already the traditional DNA sequencing was the Sanger sequencing, but now we can sequence thousands of genes at a time. So the next generation sequencing has filled that gap and become an everyday research tools to address discussion. So therefore, what can we do with this NGSs? NGS technology has fundamentally changed the kind of the question the scientists can ask and answer. So the innovative sample preparation and the data analysis option enable a broad range of application. So therefore, for what purpose we can use NGSs? We can rapidly sequence the whole genome, right? For example, we can sequence the human genome, right? The whole human genome at a time. So that's with NGSs is possible. Or we can sequence deeply sequencing the target regions. So in case if we have some target regions with NGS, we can sequence that one. Or we utilize RNA sequencing to discover a novel RNA variants and quantify or we quantify the messenger RNA for a gene expression analysis, right? And also we can analyze epigenetic factors such as a genome-wide DNA methylation and a DNA protein interaction. So we can do that one too with NGSs. And also it's possible to sequence the cancer sample to study rare somatic variants and the tumor subclonal and more. And at the same time we can use also for study the human microbes, or it could be the vector microbes. And maybe if we're lucky, we can also identify a novel pathogen with NGS. So NGS, by now most of the research are using NGS, and you guys you need to know in detail about NGS in case if you have some researches, if you have a plan to do to apply for a grant, if you have a plan to do laboratory works, so it would be nice if you know NGS for your future career, because this is just a laboratory technique that can help you to do your research. So this is just an introduction about NGSs, and maybe for next time I would like to present for you in depth about NGSs in different fields. So if you like the video, just share it and subscribe and like it, and that's it. What for today.